Hello students. Today's topic of discussion is fundamentals of genetics. And in this chapter, we are going to learn about the X-linked inheritance and monohybrid and dihybrid crosses. Now, before we go into the details, we should know what exactly is genetics. Now, genetics is the branch of biology which deals with the study of transmission of hereditary characteristics from parents to the offsprings. Hereditary characteristics, it means those characteristics which can be inherited, which can pass on from one generation to the next generation, or in other words, from the parents to the offspring. So that branch of biology which deals with the study of transmission of hereditary characteristics from the parents to the offsprings is called genetics. Now we should not be confused between genetics and heredity, because heredity is not a branch. It is the process of transmission of hereditary characteristics from the parents to the offsprings. Now, what is this X-linked inheritance? In human beings, there are 23 pairs of chromosomes. Out of the 23 pairs of chromosomes, 22 pairs are autosomes, which are responsible for determining the body characteristics, and the remaining one pair is known as allosomes or the sex chromosomes. Now, the allosomes in males, it is represented by X and Y, and in case of females, it is denoted as X and X. Or in other words, females have two X chromosomes, whereas males have one X and one Y chromosomes as allosomes. Now, X-linked inheritance means it is the inheritance of characters due to the presence of genes exclusively on the X chromosomes. That means we are going to study about certain disorders which occur due to the presence of genes which are only present on the X chromosomes. Here, Y chromosome will not be taken into consideration. So, X-linked inheritance refers to the inheritance of characters due to the presence of genes exclusively on the X chromosomes. And two such common examples of X-linked disorders are hemophilia and color blindness. Before we take up an example to go into the details and understand what exactly happens, we must know what is hemophilia and what is color blindness. Now, hemophilia is a genetic disorder or a hereditary disease in which the blood clots very slowly. This is due to the absence of clotting factors. Clotting factors are nothing but special proteins which are required for the entire process of blood clotting. And why there is an absence of clotting factor? Because the gene is defective. Since the gene is defective, it cannot express itself. And since it is not able to express itself, the protein is not synthesized. And as a result, the person suffers from hemophilia, in which the blood clots very, very slowly. This is also referred to as the bleeder's disease because a minor injury might result in excessive bleeding, resulting in his death. Now, color blindness is another example of hereditary disease in which a person cannot differentiate between colors, especially between red and green colors. This is also due to our defective gene. Now, these two examples which I cited, these are X-linked disorders. That means the gene which is responsible for causing these disorders are located exclusively on the X chromosome. Another important feature you need to understand is that these genes are recessive genes. Recessive means they can only express themselves when they are present in similar pairs, or in other words, in homozygous condition. So, two things you learned about X-linked disorders. First thing, the gene responsible for causing this disorder is located exclusively on the X chromosome. And the second thing you learned is that this gene is a recessive gene. That means it can only express itself and result in the manifestation of the disease in the individual when it is present in similar pairs. Now this slide shows the genotypes of a female and the male. These are the possible genotypes of the female. So here in this slide, we have taken up the X-linked disorder color blindness to understand the entire concept. Now X and X, when we are writing, this is the genotype of a normal female. Now here in the second one, this female has the recessive gene in the form of C on one of the X chromosomes, but the other X chromosomes is having the dominant allele, which will suppress the expression of this recessive allele that is located on the other X chromosome. So this female, she is a carrier because she is carrying the gene on one of her X chromosomes, but she does not express the characteristics of color blindness. The reason being the dominant allele is located on the other X chromosome. So she is referred to as a carrier. Now look at the third genotype in the female. Here, both the X chromosomes, they have the recessive alleles. Here, it is this X chromosome has the C gene and this X chromosome also has the C gene. So, I said this is a recessive gene which can only express itself when present in similar pairs or when it is found in homozygous condition. Since both the X chromosomes are having a pair of this recessive gene, the gene expresses and the character manifests, and as a result, this female is a colorblind female. Now look at the genotypes of the male. When I said genotype, what am I referring to? Genotype is the genetic constitution of an individual. Now, X and Y represents the genotype of a normal male. Now, have a look at this. This male has the recessive gene on the X chromosome. So, will this male be a carrier? Definitely not. This male won't be a carrier. There is no carrier option in case of males because there is no dominant allele located on the Y chromosome of the male. That means I had told you earlier this is a recessive gene located exclusively on the X chromosome. So the dominant allele cannot be found on the Y chromosome. So when the recessive gene is found on the X chromosome of the male, the character shows or manifests because the gene expresses itself and the male is a colorblind male. Such condition in males is referred to as hemizygous condition. Now this slide is demonstrating one of the many possibilities. Now here, the father is an unaffected father, which means he is a normal father. That means he has XY chromosomes. He is not carrying any recessive allele which might determine the defect or disorder. Now look at the mother's genotype. The mother's genotype, it has got two X chromosomes, but on one of the X chromosome, the recessive gene is present. She is a carrier mother. Why she is a carrier mother? The reason is the dominant allele located on the other X chromosome suppresses the effect of this recessive allele. So she carries the gene, but she doesn't express the symptoms of this disorder. Now, what is the outcome? What are the possibilities in the offsprings which are produced in the next generation? The offsprings can be an unaffected son, unaffected daughter, affected son, and unaffected carrier daughter. Let me explain this. When this normal X chromosome of the mother combines with the X chromosome of the father during the process of fertilization, then it will result in an unaffected daughter. So she is a normal daughter. Now, when this normal X chromosome in the female combines with the Y chromosome of the male, it will result in unaffected son. That means he is a normal son. He is not carrying the recessive gene. Now, what will happen when this X chromosome carrying the recessive allele combines with the normal X chromosome of the father? So this will result in a carrier daughter. So she will be carrying the recessive gene on one of her X chromosome denoted by this red color, but she won't be showing symptoms of this disorder. So she is a carrier. Now what will happen when this X chromosome carrying the recessive gene combines with the Y chromosome of the father? It will result in an affected son, or a colorblind son, or a hemophiliac son, depending on which gene is present on the chromosome. Because why the character manifests in the son? The reason is the recessive gene is present on the X chromosome of the male, but there is no dominant allele on the Y chromosome to suppress the effect of this recessive gene. As a result, the gene expresses itself and the character manifests. So, the son born out of such mating will be either a hemophiliac or a colorblind male. The next section of our discussion is about the monohybrid cross and dihybrid cross. But before that, we need to know which organism was used as the object of experimentation and by whom. Now, Gregor Johann Mendel is regarded as the father of genetics. He conducted his experiments on the garden sweet pea plant scientifically called Pisum sativum. So, the scientific name of garden sweet pea plant is called Pisum sativum. Now, most of the experiments were based on this organism. He was a monk, so he used to live in a monastery which had a huge garden. So he used to cultivate this particular plant, and he conducted numerous experiments on this plant and deduced various laws of genetics. Now, basically, he studied these 7 characteristics in the pea plant. What are the characteristics that were dealt with? Height, seed shape, seed color, seed coat color, pod shape, pod color, and flower position. Pod refers to the fruit. These were the characters, and they were in 2 different forms which were referred to as traits. So what is a character? Any heritable feature of an individual is referred to as a character. And what are traits? The alternative forms of a character is referred to as traits. For example, height is the character. The traits are tallness and shortness, or tallness and dwarfness. Seed shape is the character, and the alternative forms or the traits are round and wrinkled. For height, tall was the dominant trait, and for seed shape, round is the dominant trait. For seed color, yellow was the dominant trait. For seed coat color, green was the dominant trait. For pod shape, inflated pods is the dominant trait. For pod color, green color is the dominant trait. For flower position, axial position is the dominant trait. Correspondingly, shortness, wrinkled seed shape, green seed color, white seed coat color, constricted pod shape, yellow pod color, and terminal flowers were the recessive trait. Let me make one thing very clear over here. Dominant trait usually expresses itself in the presence of the recessive trait. Or in other words, the gene that is responsible for determining the dominant trait always expresses itself in the presence of the recessive gene. As long as the dominant gene is present, the recessive gene will remain suppressed. And the recessive gene can only express itself when it is found in homozygous condition or in similar pairs. Monohybrid cross. What is monohybrid cross? When a cross is made between 2 parents which differ in one pair of contrasting traits, then such a cross is called monohybrid cross. When a cross is made between 2 parents are taken into consideration which differ in one pair of contrasting traits. That means, let's say one parent is tall and another parent is dwarf, and the parent who is tall is a pure tall individual, and the parent who is short or dwarf, it's a pure dwarf individual. That is, so what is that contrasting traits over here? Tallness and dwarfness, or tallness and shortness. And what is the character we are talking about? We are talking about height. So when a cross is made between 2 parents which differ in one pair of contrasting traits, then such a cross is called monohybrid cross. This slide explains the definition of monohybrid cross. You can see over here, one parent is a pure tall plant which is represented by the genotype capital T, capital T. Capital T gene determines tallness in an individual, and Like chromosomes always occur in pairs, the genes also always occur in pairs. So this parent is a pure tall plant and the genotype explains that. It has the genotype capital T, capital T. And this individual is a homozygous dwarf plant because the recessive genes small t and small t are present. Small t or t in the lower case, it represents the gene which will determine the recessive trait in the individual. And I have told you earlier, the recessive gene expresses when it is present in homozygous condition or in similar pairs. So, when a cross is made between two parents which differ in one pair of contrasting traits. So, one pair of contrasting traits, how? Tallness and dwarfness. So what are the gametes which are formed out of these parents? The gametes which are formed by the homozygous tall plant will be having the capital T gene, and the gametes which are formed from the dwarf plant, from the homozygous dwarf plant, will be having the recessive gene, that is small t. When these gametes fuse during fertilization, the next generation of offsprings are produced, which are represented by the genotype capital T and small t. Now the question is, these individuals which are formed in the next generation, they are represented by the genes capital T, small t. But are they tall? Or are they dwarf? Or are they of medium size? There is no such thing as medium size. I had told you earlier, whenever the capital T gene is present along with the small t, capital T being the dominant gene or the dominant allele, it expresses itself and manifests tallness in the next generation. The recessive gene will remain suppressed in the presence of the dominant gene. So, the generation of offsprings which are produced, they are all tall plants. So, that is the phenotype of the individuals. The observable characteristics in the individuals is called phenotype. Genotype is heterozygous tall. Heterozygous, why heterozygous? The reason is the alleles are not present in similar pairs but in dissimilar pairs. One capital T and one small t. So, these are all heterozygous tall plants. This generation of offsprings obtained as a result of crossing between the parental generation is called first filial generation or the F1 generation. Now next, Mendel, he self-fertilized the F1 hybrids. These are the F1 hybrids. So the F1 individuals were self-fertilized. They were crossed. So these were the gametes which are obtained. Two gametes were formed and this gamete will be having capital T gene and here this gamete will have the small t gene. Similarly for this plant also. So likewise the number of gametes which are produced by this plant, 50% would be having the capital T gene and the remaining 50% will be having the small t gene. So when a cross is made or when fertilization occurs, there are four possibilities. Capital T combining with capital T. This is the outcome. Capital T combining with small t. This is the outcome. Small t combining with capital T. This is the outcome. You must be wondering why we haven't written small t and capital T. The reason is that priority is given to the allele which is dominant. That is why we write capital T by convention first, followed by the small t. Then the small t combines with small t and it results in this individual. So what is the genotype of this individual? It is a homozygous tall plant or homozygous dominant plant. What about these two? They are heterozygous dominant individuals. And what about this one? The last one is a homozygous recessive individual. So this generation of offsprings which are produced as a result of crossing between the F1 generation is called the second filial generation or the F2 generation. So I gave you the genotypes of the individuals. Now let us determine the phenotypes. Phenotype is the observable characteristic of an organism. This individual's genotype is homozygous tall or homozygous dominant. What would be the phenotype? Tall. What about this individual? This individual's genotype is heterozygous tall. What would be the phenotype? Tall. What about the genotype of this individual? Genotype is capital T, small t. So the genotype is heterozygous dominant or heterozygous tall. Now what would be the phenotype of this individual? The phenotype would be, it is a tall plant. And the last one, what is the genotype of this individual? It is a homozygous dwarf plant or homozygous short plant. So what is the phenotype? Short, or it is a dwarf plant. Now, if I find out the ratio for the genotypes and also for the phenotypes, so what is the genotypic ratio? Genotypic ratio is 1:2:1 because 1 homozygous dominant, 2 heterozygous dominant and 1 homozygous recessive. So, the genotypic ratio is 1:2:1. Now what is the phenotypic ratio? Phenotypic ratio is 3:1. Why 3:1? Because 3 are tall and 1 is dwarf. So the phenotypic ratio of a monohybrid cross is 3:1 and the genotypic ratio of a monohybrid cross is 1:2:1. Now, let's move on to dihybrid cross. What is dihybrid cross? When a cross is made between two parents which differ in two pairs of contrasting traits, then such a cross is called dihybrid cross. So if you are asked to differentiate between monohybrid and dihybrid cross as far as the definition is concerned, for monohybrid cross, you will be writing, when a cross is made between two parents which differ in one pair of contrasting traits. But here, in dihybrid cross, you'll be writing, when a cross is made between two parents which differ in two pairs of contrasting traits, then such a cross is called dihybrid cross. Now, this slide explains a dihybrid cross. It seems to be a little complicated, but little bit of practice and little bit of concentration will help you to overcome this fear of solving a dihybrid cross. So in dihybrid cross, we have to take two characters into consideration, which will be having 4 different traits. Let's say this parent, it produces seeds which are round in shape and the seed color is yellow. So since it is represented by capital R, so capital R is for the dominant trait. So capital R allele is the dominant allele which determines roundness in the seed. And capital Y, which is for the color of the seed, that's yellow, it is also the dominant allele. So what are the corresponding recessive traits? if capital R is determining roundness of the seed, then small r is determining the wrinkleness of the seed. So, round is the dominant trait, wrinkled is the recessive trait as far as the shape of the seed is concerned. Now, color of the seed or color of the seed coat, if it's yellow in color, then it is the dominant trait. If it is green in color, which is represented by small y, then this is the recessive trait. And these two individuals represent the parental generation. So this parent is homozygous round and homozygous yellow. And this parent is homozygous wrinkled and homozygous green. So they are pure for the characters we have taken into consideration. Now in the next generation, after the gametes are formed by these individuals, the next generation of offsprings, all of them will be having the genotype capital R, capital Y, small y. So, this generation of offsprings which are produced as a result of crossing between the parental generation is referred to as F1 generation or the first filial generation. And all the individuals which are formed during the F1 generation, they are heterozygous round and heterozygous yellow. Now, that is the genotype of these individuals. If we are asked to write the phenotype of these individuals or of the F1 hybrids, then we are writing they are all round and yellow. That means the individuals which are formed in the next generation, they produce seeds which are round in shape and yellow in color. Now, Mendel, he self-fertilized the F1 generation, self-fertilized the F1 hybrids in the next generation. So, from each parent, we are getting the gametes. 4 types of gametes, in fact, are produced by each parent. What are the 4 kinds of gametes? Capital R, capital Y, capital R, small y, capital Y, and small r, small y. So, if this is the genotype of one F1 hybrid, how do we get the combination? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. This is how we get the gametes of one parent. Similarly, on the other side, we are writing the gametes of the other parent, which would be the same as that of the first parent. Because here in the next generation, 2 F1 hybrids are self-fertilized. Now, in order to get the possible combinations, we have to draw a square. This square is referred to as Punnett square. Punnett square is a square in which the gametes of one parent are written along one side and the gametes of the other parent are written along the other side, and the possible combinations are written in the subsquares. So we have obtained the possible combinations. How do we get it? For example, let us consider the gamete, this gamete, having the genes capital R, capital Y. So this gamete having these genes will be fusing with this, or it might fuse with this, or it might fuse with this, or it might fuse with this. So these are the resulting individuals which are formed. Similarly, for the second one, this can combine with this, or with this one, or with this one. So we get this one, all the individuals having the genotypes. The third gamete, this might combine with this, or with this, or with this gamete, or with this gamete, resulting in the production of these individuals. Finally, this gamete again might combine with this or with this or with this gamete and finally with this gamete, resulting in the formation of these individuals. So if we are determining the phenotypes, by seeing the genotypes, we got to determine the phenotype because phenotype is genetically controlled. It is always determined by the genotype. So this individual will be round and yellow. This will also be round and yellow. Why it will be yellow? Because capital Y gene is dominant over the small y allele. This one will be round and yellow because capital R allele is dominant over small r allele. This one will be round and yellow again. What about this individual? This will be round and yellow. But this one will be round and green. Why green? Because the small y, which is responsible for greenness, it is present in homozygous condition or in similar pairs. So this individual will be round and green. This again will be round and yellow. This one will be round and green again. This will be round and yellow. This will be round and yellow. This one will be wrinkled and yellow. Why wrinkled? Because the small r gene is responsible for determining wrinkledness in the seed. So, since it is present in similar pairs or in homozygous condition, the seeds will be all wrinkled in shape but yellow in color. This is also wrinkled and yellow. This is round and yellow. This is round and green. And finally, this is wrinkled and yellow. And the last one, it has wrinkled and green characters. So, at the end of crossing of this generation, we obtain 16 individuals. And out of 16 individuals, 9 of them are having the characteristics round and yellow, 3 of them round and green, 3 of them wrinkled and yellow, and 1 wrinkled and green. So, what is the phenotypic ratio of the F2 generation or the second filial generation? It is 9:3:3:1. So, if you are asked what is the phenotypic ratio of a dihybrid cross or of the F2 generation of a dihybrid cross, you are writing it is 9:3:3:1. And what is the genotypic ratio of a monohybrid cross? It is 1:2:1. And what is the phenotypic ratio of a monohybrid cross? It is 3:1. We can find out the genotypic ratio of a dihybrid cross, but it would be very long and it is very complicated. So I hope you have understood the topics which I have discussed today with you. First, your X-linked inheritance, explaining that hemophilia and color blindness are X-linked disorder which occur due to the presence of genes exclusively on the X chromosome, and these genes are all recessive genes. And the second concept which you learned is about the monohybrid cross, and lastly about the dihybrid cross. Thank you, children.